Hey everybody, in this video I am going to talk about a relatively new technique for molecular biology. Now this is called CPD-seq for CPD sequencing, and it was invented by researchers in 2016, so not too long ago. And what does it stand for? So it stands for cyclobutane pyrimidine dimer sequencing. Now, what is a cyclobutane pyrimidine dimer? So essentially, this involves UV light, and UV light can damage DNA. And what it does is it can form covalent bonds between adjacent pyrimidines. So let's say we have a molecule of DNA where there are two T's side by side on one strand of DNA. So we can get covalent bonds formed between those nitrogenous bases. Same thing for CT. T can be on the 5' side, C on the 3' side. Again, we can get covalent bonds formed between those. Also between two C's. Now, I'm going to show you some structures in a little bit, and we'll see where this cyclobutane comes from. But essentially, when these covalent bonds form between the nitrogenous bases, it causes problems. It causes problems in the DNA. So I have a conceptual diagram here. So let's say we have a section of DNA with two thymines, hydrogen bonded to two adenines. Let me connect these guys down here. We have one DNA strand up top. The 5 carbon sugars are the squares, 5' to 3', opposite orientation down here. Now, a cyclobutane pyrimidine dimer, what would happen is we would get covalent bonds formed between these two thymines, and it might look something like this. So here I'm showing the covalent bonds forming, and again I'm going to show you a more accurate diagram in a bit. But what we can see when this forms, it disrupts the structure of the double-stranded helix, right? So that's what we're showing you with what I'm showing with this little kink right here. And we can't get proper hydrogen bonding between here, and this structural damage causes a number of problems for the cell. For one, if this top strand was the template, well, it couldn't be used in transcription. Another thing, if the cell was going through replication, and let's say this top strand was the template, well, DNA polymerase couldn't use these T's as the template. And if it tried to use these T's, it might put in the wrong base, because hydrogen bonding, proper hydrogen bonding doesn't work when we have these covalent bonds formed between these adjacent T's. And it could be a C and a T or two C's, like I already said. So the cell has a process called nucleotide excision repair, whose job it is, among removing other types of DNA lesions, one of the most important jobs is to remove these CPDs. So again, what is NER? This is nucleotide excision repair. And make sure you check out the other video. I'm going to have another video that goes over this mechanism in detail. In this video, I'm going to mostly just focus on CPD sequencing because I think it's really cool. I haven't actually put this video on nucleotide excision repair together yet. I'm going to do this one first, and hopefully I'll order them. So you'll cover this first in the videos, and then see this video I'm preparing right now. So I've always been really interested in NER because, this is something we all have to worry about, right? So nucleotide excision repair, why is it important? Well, it repairs UV damage. So it repairs those pyrimidine dimers that form in our skin cells if we should be exposed to sunlight or the UV rays in the sun. And that's why we wear sunscreen when we go outside, or we should wear sunscreen if on exposed skin. So why do we know it's so important? Well, there are people with a disorder called xeroderma pigmentosum. And if you took genetics with me, we talked a lot about this disease, the complementation groups associated with this genetic disorder. So these individuals really can't go in the sun at all. They can't go in the sun at all, and they have really high rates of skin cancer. And what they're lacking is their cells are lacking the ability they're lacking nucleotide excision repair. They're lacking a protein needed to properly perform nucleotide excision repair and remove cyclobutane pyrimidine dimers. So NER is important. NER allows us to go outside. So these XP individuals with XP can't even really go outside. So NER allows us to go outside. So pretty important mechanism, right? So knowing that these individuals exist and knowing what they have to go through, not being able to go out, even just go out inside in the sun, even with sunscreen, really not supposed to do it at all, leads me to think, what about us? And unless I'm assuming none of my students in the class have XP, I don't know. I think I would know. But what if we were to go outside? What if this is me right here? I've got all my skin covered with clothing, but let's say my arm right here. So this is just a regular old sunny day. Sunlight on my skin. We look at my skin layers right here. So I haven't had anatomy and physiology. I haven't had a course like that in 20 years. So forgive me if this isn't very accurate, but okay, top layer of skin cells, dead, right? And then we go a little lower, and then we've got some live cells in here. So I've always wondered, let's say you're in the sun for 1 hour without sunscreen. Now, each of these cells, we have like our genome, and each of these cells right here, they say these are the nuclei. How many CPDs are forming per hour in each cell? Is it one? Is it hundreds? Is it thousands? I've always been curious about that. And I think that there are some older methods that have been used to estimate the rate of CPD formation in skin cells. And I think these studies have been done by having volunteers go out and taking skin biopsies. But this new technique, CPD sequencing, seems to be a very powerful way to study things like this. And it hasn't, as far as I know, it hasn't been applied to humans yet. It's mostly used so far as I know, for studying CPD formation in microorganisms. So I really like this technique, and let's take a look at how it works. And then, eventually, I'm sure it'll be used to study CPD formation and nucleotide excision repair in humans. So, okay, what is CPD-seq? What does it do? Essentially, it finds all the CPDs in the genome, or a genome, or a collection of genomes. So, for example, the technique, as I've seen it, has been used to study yeast. So Saccharomyces cerevisiae, budding yeast. Let's say we take 1 million cells, and then we expose them to UV light for a certain amount of time. Then, after exposing them to UV light, what we do is we get the genomic DNA. Well, we wait. We either wait 0 hours, 1 hour, or 2 hours. When I say wait 0 hours, that essentially means you're not waiting. So right after the UV exposure, you're going to do something. For this one, we're going to wait 1 hour before we do the next thing. For this one, we're going to wait 2 hours. And so the next step is to isolate the genomic DNA. So by waiting for 1 hour or 2 hours, we've given the yeast cells a certain amount of time to try and fix any CPDs that formed as a result of exposure to UV light. So once we have the genomic DNA for each of our samples, what we do is we would share the DNA into fragments. Not share DNA fragments, share DNA into fragments. And let's say those fragments are all about, 500 base pair to 1,000 base pairs long. And what I'm going to do is, so we'll have millions, right? We have millions of yeast. And I forget how big the yeast genome is. I'm not going to guess, but millions of base pairs in the yeast genome. And so when we're shearing all those DNA molecules into fragments, we're going to have millions and millions of these fragments here. And let's say one of these fragments right here has a CPD formed between these two adjacent thymine residues. So that's what I'm going to do here. I'm just going to use this one molecule to represent the millions of molecules that are in our sheared fragments in each of these samples, 0, 1, and 2 hours. So shearing means break the chromosomes into smaller fragments, and then we'll polish the ends so they're blunt-ended. And there are ways we can do that with certain enzymes. So there'll be a 5' end here, OH group here, 5'. The 5' ends have phosphates. OH group here. Now the next step is going to be, let me get a new piece of paper here, quickly diagram, make sure I'm still on camera. And if you're watching this on YouTube, sometimes you'll be watching in standard definition, and you can usually click in like the bottom right of the video and switch it to high definition, which might make it easier to see some of the things I diagram down here, some of the smaller characters I use, letters or drawings. So now this OH represents the 3' OH, right, of a 3' nucleotide, of a nucleotide at the 3' end. And the P represents the 5' phosphate group on the 5' end. So the next step, after we have all these fragments from across the genome, some of them are going to have permitting CPDs, some won't, is we're going to add these things called adapters. And the adapters essentially are just little DNA molecules, 2 strands of a known sequence. Let's say, I don't know exactly how long they are, but let's say they're 100 base pairs long. I'm going to color these green. Okay, so what we're going to do is stick these onto the ends of these DNA molecules with DNA ligase. So let me put these adapters here. Let me see, maybe I'll color it in later. So the adapters are going to be ligated to the ends on both sides. Now we have a 5' phosphate here, OH here, OH here, phosphate down there. Next step, we are going to add DDATP. Now some of you might remember this stands for dideoxy. We're going to use this with an enzyme called terminal transferase. And what this is going to do, this enzyme will do, we'll take this and it will add this ATP to the 3' ends, any 3' end that has a hydroxyl. So it's going to add one adenine nucleotide. But that adenine nucleotide is going to be lacking a 3' OH group. So we're essentially getting rid of these OH groups by adding an A nucleotide without a 3' OH. And that's going to be important. So instead of an OH now, I'm going to put an A, and I'll put 2 Ds for dideoxy. Same thing on this side, A dideoxy. Looks like I said add here, but that's A, and then DDs for dideoxy. P for the 5' phosphate. Now, the next step, and this is kind of why I wanted to, another reason why I want to talk about this technique is that it brings in enzymes from base excision repair. So the technique is used to study nucleotide excision repair, but it involves enzymes from base excision repair. So here we're going to add an N-glycosylase. So make sure you go back and watch the base excision repair video if you don't know what an N-glycosylase is. The specific one we're using is T4 endonuclease 5, Roman numeral 5. And this is specific for CPDs. So 2 T's that have the covalent bonds between them, or CT or TC or CC. So that's what these are the substrates for this DNA N-glycosylase. Now, this is where I wanted to look more in detail at the structure of a, let's say, a thymine dimer. So a thymine dimer would be a CPD formed of 2 thymines. So let me quickly diagram here, and maybe I'll speed up the video so you don't have to watch me diagram this. So far I have diagrammed 2 thymines. So this is one, and it's probably in the reverse, it's probably flipped to how you're used to seeing them. And the reason why I wanted to do that is I want the T's to be up top and the A's to be down bottom here. So this is where the A's, the adenines would be, that these would be hydrogen bound with in a molecule of DNA. Now, for those of you who have had organic chemistry, and I think that's a prereq for this course, you can see this is the cyclobutane. So these are the bonds here, the covalent bonds formed between the 2 thymine residues that aren't normally here. And also there are going to be 2 methyl groups here. But this 1, 2, 3, 4 butane is like a 4 carbon chain or 4 carbon ring. Cyclobutane is a 4 carbon ring. We have it here, 1, 2, 3, 4. Okay, so that's where that cyclobutane comes from. Now, these are the nitrogens that are attached to the 5 carbon sugar. So I'm going to diagram the 5 carbon sugars, and I'll speed up the video. Okay, everyone. So here is a more detailed diagram of essentially, what is a more detailed diagram of? These are the two thymines. This is the cyclobutane that formed between them. So we're looking at these two thymines, two sugar molecules. Two thymines, two sugar molecules. So, and let me show you one more thing. Back up a little bit. We're going to add Nglycosylase, and Nglycosylase is going to process these DNA molecules with the pyrimidine dimers, right, with the CPDs. So what is it going to do? Well, this is an Nglycosylase that has both an Nglycosylase activity, which means it's going to separate this bond right here. This is the one it targets, not this one. And when it does that, this T4 endonuclease 5 also has a lyase activity, and we talked about some of those in the base excision repair video. So it's going to cut here, and it's also going to cut here, and it's also going to cause a break here. So essentially what's going to happen is, let me see, do I have my eraser here, is we're going to get rid of that. We're going to break this, and we're going to break that. So it's going to separate this thing right here from this piece right here. And I should also say, because this is broken right here, we're going to get some funny thing here. I don't know exactly if this is what, this is an aldehyde. I don't know exactly if this is what it looks like, but conceptually we see that this 5-carbon sugar is no longer a ring. It's no longer this 5-carbon ring here. It's got a funny structure because of the way that the glycosylase broke these bonds here. And I have a link to actually a really good website where you can see all the way the bonds are really thought to move and break and things like that. But this is good enough for now. So essentially, this is what's going to happen right here. And I can diagram that with a more conceptual, in a more, I guess, just conceptual way, like so. Let's see. So now I'm going to put this as that weird aldehyde, and then that T that used to be here is now going to be, ooh, I know what I want to do. Sorry. Okay, I need to show you a cleaner diagram to explain what's going on here. So I'm going to use this one right here. So remember, before that Nglycosylase broke the bond between the nitrogenous base and the 5-carbon sugar, it looked something like this. So Nglycosylase comes in, the T4 endonuclease 5 comes in, breaks this, cuts one of the bonds in here, and breaks that too. So we get something like this. So essentially separating this part, no more covalent bonds between these things over here and these things over here. In that T, it's now covalently linked to this nucleotide over here. So it's stuck on that strand over here. So I hope that makes sense, or some sense. So, okay, I'm gonna finish this diagram now. And this piece up here, right here, I'm gonna indicate that just like that right here. Okay, so at this point, it's been treated, these molecules have been treated with Nglycosylase, and remember, this is just representative of millions of sequences that have CPDs in them. And the CPDs have been broken by T4 endonuclease 5. You can color these adapters in. So we're going to add another enzyme now that we also saw in base excision repair, AP endonuclease. Now essentially what AP endonuclease is going to do is it's going to remove that broken 5-carbon sugar there, that broken ribose, or deoxyribose. It's going to get rid of that, and it's going to leave a 3' OH group on that nucleotide right here. So remember what this DNA molecule used to look like, right? ATTA. You know, after treating with T4 endonuclease 5 and AP endonuclease, now what it looks like is this. With this T linked here, this is going to have a 5' phosphate right here and a 3' hydroxyl on this A. So let me diagram that quickly for us. Just going to put an OH right there. And why not color those in? You know, if you have no clue what's going on right now, you can at least enjoy the pretty colors. So, okay, now at this point, we're almost ready to figure out where all the CPDs are in the genome. So now we're going to denature all these molecules. And then what we're going to get is something like this. I'll color these in here. So each of those molecules we looked at is going to produce 3 different DNA molecules after denaturing, right? We're going to have this one, we're going to have this one over here, and we're going to have this one right here. So each of those double-stranded molecules after denaturing will produce 3. Note that only one of these fragments has a 3' hydroxyl group on the 3' end. Oh, the 3' nucleotide on the nucleotide at the 3' end. Only one of them has a 3' hydroxyl on that nucleotide. So this molecule here has the dideoxynucleotide at the 3' end, and so does this one over here. So the process of CPD-seq uses the fact that only these molecules have this 3' hydroxyl here to its advantage. So let's see here. I think I want to use this color. So at this step, another DNA adapter molecule is going to be added to all DNA molecules with a 3' OH group at the end. So let me say this is going to be about 80 base pair, 80 nucleotides. This is single-stranded, so all these DNA molecules are single-stranded. And so DNA ligase will be used to ligate the 5' end of this adapter molecule to this end down here, and there's probably no OH group here. So after that's ligated, all of these molecules with a 3' OH group are going to look something like this. So a single-stranded DNA molecule with a green adapter on one end and a purple adapter on the other end. So at this point in the process, the tubes are tubes from the 0 hour time point and the 1 hour time point and the 2 hour time point are going to have millions of these fragments right here. And the next step is to load those into, and they need a little more processing for this to work, but essentially load them into a high throughput sequencing machine. High throughput sequencing machine. Sequencer. And a common company that makes these machines is Illumina. So these sequencing machines can sequence millions of DNA molecules at the same time. And then when they're done, we essentially get a giant text file with each DNA sequence. And so we can sequence all of these molecules here, and then we can use computers to remove any sequences that match this green adapter and this purple adapter. And then we're left with like millions of these guys right here, millions of sequences, each one corresponding to the DNA between a green adapter and a purple adapter of a DNA molecule. And so what would we do then? Well, then what we do, so I'm going to just focus in on one of these. So this is an example sequence that might be found between a green adapter and a purple adapter. So at this point, because we're working with yeast, what we would do is go to the yeast genome sequence. And yeast has 16 chromosomes, and as an example, I diagrammed 4 different random chromosomes here of yeast. We would search the yeast genome sequence for this sequence right here. Let's say we found this on chromosome 4. And so I'm zooming in to this little section of chromosome 4, and I'm looking at the DNA sequence here. And we should be able to see, I don't think I diagrammed the whole thing, but let's see, G-A-A-T-A-T-T, G-A-A-T-A-T-T. Okay, so I found it starting right here. So we're looking in this chromosome, the sequence of this chromosome, and we're going to go all the way to GGAG. Okay, so here is the sequence in the yeast genome that matches the sequence between these 2 adapters. Okay, so what does that mean? Well, that means these 2 T's right here, these were the 2 T's that formed a CPD during our experiment. And because they formed a CPD, this sequence here ended up between these 2 adapters. So that's essentially how the technique works. And because we have millions of these sequences, we can find where the millions of CPDs are throughout the yeast genome. So at this point, I hope you understood that. It's a very complicated process, but we've seen a lot of the steps needed to perform this technique over and over again, or not over and over again, but a couple times in this course. So what I suggest you should do now is go to the lecture notes and check out the 2 problems on CPD-seq, or 3 problems on CPD-seq. I think they